So welcome and everyone, and thank you to those joining us live and to those listening in afterwards. I'd also like to warmly welcome our guest today, whom I'll introduce shortly. So I'm Donnacha Fitzgerald, moderator of today's event on behalf of the FIP Technology Forum. FIP's vision is a world where everyone benefits from access to safe, effective, quality, and affordable medicines and health technologies. FIP's vision, so the mission is to support global health by enabling the advancement of pharmaceutical practice, sciences, and education. We're very pleased to be delivering this event, Precision Medicine, the Current State, the first episode of a new Precision Medicine webinar series, co-organized by the FIP Technology Forum and the FIP Special Interest Group on Personalized and Precision Medicine in the Board of Pharmaceutical Sciences. Just some announcements before we begin. Please note that this event will be recorded and made available at www.fip.org. To all those listening, please feel free to send your questions through the Q&A box, and we will be picking those up throughout today's session. So, precision medicine, sometimes known as personalized medicine, is an innovative approach to tailoring disease prevention and treatment that takes into account differences in people's genes, environment, and lifestyles. The goal of precision medicine is to target the right treatments to the right patients at the right time. With the era of precision medicine on the horizon, accompanied by a rapidly evolving technological landscape, it has never been more important for the pharmacy community to reflect on their role in its development and implementation. Our aim with this series of webinars, which we started planning at the beginning of this year, is to bring the pharmacy community closer to the precision medicine field and to identify gaps and opportunities for pharmacists and pharmaceutical scientists to fill through research and practice development. The first webinar, The Current Status, which is on today, aims to discuss the current state of precision medicine in practice, the technologies behind precision medicine deployment, and the remaining challenges and opportunities. The second webinar, Where is the Technology Heading?, will highlight some key technological advancements arising in screening and computational methods likely to shape the future of precision medicine. The third webinar, What Will It Cost?, will aim to discuss a key barrier to the implementation of precision medicine, its cost, and what is needed to overcome this. The dates for webinar 2 and 3 will soon be announced. We may also continue with further webinars in subsequent years. So what are the learning objectives of today's webinar? Well, first, we want to introduce you to the needs, goals, and principles of precision medicine and provide some examples of current implementation across diseases. Also to discuss the safety challenges and limitations of current approaches and opportunities for further development. And lastly, we are also interested in discussing national and international coordination efforts, which are crucial to the widespread deployment of precision medicine approaches. So just to shortly introduce you to our panel, which I will properly introduce later as we go through each talk. So Stephanie and I are the co-chairs and organizers of this event series. And also today we have guest speakers, Dr. Mark Dunnenberger and Einar Moltzen. Just as an overview of today's program, firstly, we'll introduce you to the webinar series, which I've just done. Next, to give an overview of the Clinical Pharmacogenetics Implementation Consortium, which is in the US, and Clinical Decision Support Systems. This is by Dr. Mark Dunnenberger. Then Dr. Stephanie Huang will provide examples of precision medicine approaches in cancer. And Einar Moltzen will give an overview of the International Consortium of Personalized Medicine and International Coordination Efforts in Precision Medicine. And lastly, we will have a brief panel discussion and Q&A. Now, first speaker is Dr. Mark Dunnenberger. So Dr. Dunnenberger is Director of Personalized Medicine and Pharmacogenomics in the Mark R. Niemann Center for Personalized Medicine at North Shore University Health System, and leads the various clinical pharmacogenomics implementation projects within the center. The goals of these projects is to improve the care and health outcomes of North Shore University Health System patients through pharmacogenomics-based strategies. He joined North Shore in 2014. Now, Dr. Dunnenberger leads the pharmacogenomics clinic which serves as part of a system-wide initiative building upon the health system's strong foundation in genetics and molecular medicine. The clinic is among only a few across the country and the first of its kind in the Chicagoland area that preemptively screens patients for responses to medications based on genetic makeup. His clinical expertise and research interests include translating pharmacogenomics into clinical practice, developing information technology solutions for delivering genetic information to both clinicians and patients in a practical manner, and evaluating methods for educating healthcare professionals about pharmacogenetics. Dr. Dunnenberger has co-authored numerous original publications, including multiple CPIC guidelines, so Clinical Pharmacogenetics Implementation Consortium guidelines. He earned his PharmD from University of Tennessee, Health Science Center, College of Pharmacy in 2012, completed PGY1 Pharmacy Practice Residency at Mission Hospital in Asheville, North Carolina, and PGY2 Clinical Pharmacogenetics Residency in the Department of Pharmaceutical Sciences at St. Jude Children's Research Hospital in Memphis, Tennessee. And now I'm very happy to hand the floor over to Dr. Dunnenberger to discuss Clinical Pharmacogenetics Implementation Consortium and Clinical Decision Support Systems.
Great, thanks for the introduction. Thanks to the organizers for the invitation to speak today. Over the next 20 minutes, I'll give a pretty brief introduction to CPIC, and then we'll touch on different types of CDS and what do you need to have CDS done. It's a very light treatment of CDS that you could do this for hours and hours and hours. It's a really rich topic. And I'll conclude with some data from what's happening at my institution today, where we've been practicing preemptive clinical pharmacogenomics for more than 7 years. If you can go ahead and advance to the next slide, please. So CPIC, or the Clinical Pharmacogenetics Implementation Consortium, is a group, and their main goal is to write guidelines to tell clinicians how to use pharmacogenomic data, if it's available, to optimize drug therapy. One key point about these guidelines is it does not answer the question about whether tests should be ordered. It's the assumption that you have the data, and now the group is going to optimize and help you optimize drug therapy using that data that you've already collected. If you go to the next slide, please. So since last summer, there are more than 400 members in CPIC. It's clinicians and scientists. It's not just the United States. It represents 35 different countries. There are some observers that are not active members, but they're overseeing what we're doing and watching. Most of these are federal or national regulatory organizations. And I'm going to spend a little bit of time talking about a subgroup in CPIC called the informatics group. And this is a smaller group, but really important for clinical decision support. If we can go to the next slide, please. So here's a map where you're seeing where everybody is from. Big takeaways here, right across the world. Both industry and academic and health system members are here. Obviously, based on this map, heavily representing the United States, but we are getting more global representation. And you'll see as guidelines get updated, we're including more international authors to bring in perspectives outside the United States. So it's an area of interest to you and you have some expertise. Join CPIC, and there's always a call for authors, and throw your name in there, and there's a vetting process, and hopefully you can get on a CPIC guideline at some point in the future and bring perspective from wherever you're practicing pharmacogenomics. We can go to the next slide. Okay, so some key points about CPIC guidelines. Again, based on the assumption that you have pharmacogenomic results, not whether you should do testing or not. They're all in a standard format. Similar to other clinical guidelines, they have a grading of evidence and recommendations that is standard throughout. They are peer reviewed. They are freely available. You can PubMed them. They're recognized as guidelines on PubMed. They are updated on a regular basis. There's clear authorship with conflict of interest policies, and we closely follow the Institute of Medicine practices for quality clinical guidelines. And there actually was a paper written comparing the CPIC process to this Institute of Medicine practice, showing where we're similar and where we're not. So these guidelines are really well vetted and should be treated as a source from the experts of how you should use this information is a great jumping off point for implementations. You can go to the next slide. So what have we written guidelines on? Well, I've written a couple of these, but the group has written 26 guidelines covering 22 different genes and in a variety of disease states. Obviously, CYP2D6 is one of the most common genes written about, as it does metabolize a number of drugs, and it is very important in pharmacogenomics. But pick your disease state. There's probably a drug gene pair in there. Go read the guidelines. You can find them at cpicpgx.org/guidelines. We can go to the next slide. All right, so the guidelines are written to support this pharmacogenomic translation process. And that is to go from genotype to haplotype to diplotype to phenotype to therapeutic recommendation. The genotype all the way down to diplotype is covered in the supplemental material. The diplotype to phenotype is covered in table 2 of the guideline, and phenotype to therapeutic recommendation is covered in table 2 for any given guideline. I'll show you an example of those tables in a minute. But this process of pharmacogenetic translation is one that anyone who practices clinical pharmacy should be comfortable doing, because it's going from a laboratory value, which would in this case be a genotype and or diplotype, and then turning it into a calculated value, in this case phenotype, and then making a therapeutic recommendation. The most common analogous process to this is thinking about how do you renally dose adjust a patient based on their serum creatinine. So serum creatinine is the laboratory-based value. You calculate a creatinine clearance, that's your calculated value of your phenotype, and then you use that creatinine clearance to apply the therapeutic recommendations, such as dose reduce or lengthen out your interval of your antibiotic. So this process, while it's the words are different, it's very familiar to people who practice clinical pharmacy, and everyone who is a pharmacist should be comfortable doing this type of thing. And CPIC is oriented to help you do it for pharmacogenomics. Okay, if we can go to the next slide, please. So this is an example of table 1, where we're going from, on the right-hand side, the different diplotypes, and on the left-hand side, you have the phenotypes. And so you'll find this in every CPIC guideline. This is a simpler one with CYP2C19, but one of the key tables in every CPIC guideline. If we can go to the next slide, please. So here we have table 2, which is going from phenotype on the left-hand column to the third column, where we have the therapeutic recommendation. On the far right, you can see the different classifications of the recommendation. So how strongly do we feel about the evidence and whether it supports the recommendation? So this is available in every CPIC guideline. The authors spend a lot of time honing in on this wording and try to be very, very precise with these types of recommendations. So if you're ever looking at a CPIC guideline, always go look for table 1 and table 2. That's going to help you implement. And then everything else in the guideline is supporting these 2 tables and thinking about how do you actually bring this to practice. We'll go to the next slide, please. So when we think about implementing a guideline, there are 3 groups that we have to think about, right? There's CPIC that wrote the guideline. There's the group of implementers, the people who are implementing the guideline, and then CPIC informatics. So let's go to the next slide and think about the implementers real quick. So if you go on the CPIC website, there's a tab labeled implementation. And what you'll see here is a list of institutions who have volunteered to say that they're implementing pharmacogenomics. And most of them either have a website and or a contact information from somebody who is doing that implementation. And you should feel comfortable reaching out to these individuals to learn more about their implementation. I know I'm on this list, and I know many of these other individuals, and we're all generally very happy to talk about how we implement and help others implement, both in the United States and across the world, including at Children's Cancer Hospital in Egypt. Mohammed Negi, he actually came to my institution from this list and spent about 3 days learning what we were doing at North Shore and has since went back to Egypt and partnered with a number of other people and has a really great program. So quite proud of Mohammed, and was really happy to see this resource work. So there are implementers out there doing this. This is a way to contact them and learn from them. So if you're interested, feel free to do that. We can go to the next slide. So getting back to this, so you've got CPIC, which is the guidelines, you've got the implementers, and they come together in the informatics space to talk about, well, what resources can you create to help us to drive from this written document to what an implementer needs in a computable world. We can go to the next slide, please. So a growing interest in informatics for the guidelines, right? How do you take this written document, make it computable, make it scalable? And the working group comes together to have discussions about how they're using the tools that we have and what are the new tools that we need to create. And the goal is to support the adoption of CPIC guidelines by identifying and resolving where possible potential technical barriers to implementation of the guidelines with a clinical electronic environment. And so the idea here is the technical barriers shouldn't be what prevent you from implementing pharmacogenomics. It's tough enough for other reasons. So let's let this group do what it can to smooth out the clinical barriers. If we can go to the next slide. So one of the key documents that the informatics group puts together with every guideline are these implementation workflows. And basically they walk through what are the pieces of data that you need to think about and where are the clinical decision support tools that you might want to put into place. And then for each of these clinical decision support tools, there is a suggested or a starting point as to what that wording might be, what it might need to look like. So this is an example of a workflow for HLA-B 5701. So we're going to say, has the result been obtained, right? And every CPIC guideline is yes, that's where it starts. So you have the result, you put it in the medical record. The third box here on the top is put some interpretive comments related to that result. Then you need to flag it as whether it's abnormal or not abnormal, a little bit of a decision flow here. If it is abnormal or a high risk one, then you're going to go via two different pathways. Is the patient on the drug? Okay, do something about it. If they're not, have a system ready to go and fire post-test CDS if needed. And we're going to spend a lot of time talking about post-CDS here in a minute. So this is one example of what the group does. We'll go to the next slide. The biggest thing that the group has done recently is we have put the CPIC knowledge into a database that you can get off of GitHub. And the great thing about this knowledge base is it's computable and machine readable, and it's all of the supplementary material. So all of those translation tables that I talked about earlier, it's got all of the table 2 information and all the table 3 information in a computable downloadable format. So you can get all of the CPIC information at one time. Things to remember here is it's a starting point. You shouldn't take the data that comes out of the database and just immediately put it into your environment. It is the beginning, and you should review it and curate it based on your current clinical practice or your institution so that it's meeting your needs, because your needs are going to be specific to your given environment. So really important to think that this is not the end state, but just the beginning of your implementation. But it takes you from step 1 to about step 10 of a 15-step process, as opposed to without this resource, you're starting at step 1. So it speeds you up, a great accelerator, but definitely not the end point of the process. Okay, next slide, please. So what is CDS? What types of CDS exist, right? That's kind of the question that I'm going to pose in this slide. And this is oriented to the clinician-patient relationship to show one really big thing here, and that is we use a lot more clinical decision support on the provider side than we do on the patient side. And I want to pose to everyone here, is that the right approach? Is the value of pharmacogenomics wholly driven on the clinician side, or is it equal weighted for the clinician-patient side? And if that's the case, why don't we have more clinical decision support for the patient? And I would argue that's really the challenge that we have as a field going forward, is how do we balance this relationship out? But what are the types of CDS that are out there? I'll highlight a few, right? There are order sets where you're changing which medications are presented to a clinician. There are interruptive alerts, which we all know very, very well. There are interpretive comments. There are documentation tools where you're going to quickly provide a summary of the clinical meaning of a result in an automated fashion when that result comes in. You might have targeted patient notifications. So this could be an icon that tells you this patient has pharmacogenomic results. There could be a portal that you need to log into. There are a lot of different types of clinical decision support out there. And which one is best for you is going to be dependent on the situation that you're in and the resources that you have around. If you learn nothing else from what I'm saying today or take nothing else away, interruptive alerts are not the only way to do clinical decision support in pharmacogenomics. And if you only rely on interruptive alerts, you're not maximizing your value. And I'll show you some data to support that in a minute. We can go to the next slide, please. So this is what an interruptive alert looks like. The wording that you need to put in your interruptive alert should be tailored to your institution. This particular example is from St. Jude Children's Research Hospital. It's from a publication in 2013. I can tell you in my institution, this is too many words. They are not okay with having to read all of this text. They want something shorter. So you really do have to hone into your institution, but every alert, if you're going to do it, should be actionable. And that's what you're seeing in the bottom half of the screenshot. We can go to the next slide. So this is an example of a documentation tool or passive CDS. So you might hover over a CYP2D6 *1 3 lab result, and this is going to give you some more information about what that means. And cool things here are this is scalable, right? You get a really short answer, then you get a longer answer, and then at the bottom here you have a link out to a much longer answer if somebody is interested in doing this. Again, this is from St. Jude, published in 2012. We'll go to the next slide. So if you're going to do clinical decision support, what does it take? Well, it really takes 5 different components, right? You got to have genetic testing because you need to start somewhere. You need to have education because people need to understand how to use the tools that you built. And in the middle, you have to have the machinery or the ingredients to make this work. So you need to have a place to look at the genetic results, right? You get the test results, you need a discrete place to put them. You need a knowledge base or a CDS that translates these results into something meaningful. And then you need to have a GUI or a graphical user interface. You might think about this electronic medical record, but I would argue that we should go bigger and better than that today. And this is to display the results to a patient or a provider. And you need to display it in a number of different ways that meets their true clinical need at the time they're seeing it. We'll go to the next slide. So one thing that I want to drive home here is, at least in the United States, we have this problem of lab reports coming in as a PDF. A PDF is not a way to drive clinical decision support. You need to take the information out of the PDF and put it into a discrete, structured, standardized form. And that is the foundation of CDS. And you need to think long and hard about your approach to this and how are you going to store your data in a reasonable and scalable format going forward. So just remember, PDF is not good enough if you're going to drive clinical decision support. Next slide, please. I know I've said this a couple of times already, but right, the CDS that you're going to use changes across the patient journey and what's available where. And so what you're seeing here is a lot of different types of CDS. The blue and the orange is the idea of blue being physician facing or clinician facing and orange being patient facing. And again, I'm just trying to drive home here in this visual that we are underutilizing the patient clinical decision support in the patient journey. This is their data. They own it. They need to have control over it, and they need to have a good understanding. And the field needs to do a better job of building clinical decision support tools to help patients understand their results so that they can improve and be engaged in their care going forward. This is from a recent publication in CPT. So if you want to learn more about this, dive into that paper. Next slide, please. So where do I practice? I practice at North Shore University Health System. It's a community health system serving the northern suburbs of Chicago, Illinois. For the United States, it's a medium-based healthcare system. It's 5 hospitals. We're growing. Will be 8 hospitals soon. And key to my implementation is that we're an integrated inpatient and outpatient EHR, and we've been doing that for about 20 years now. We'll go to the next slide, please. So at North Shore, you can get pharmacogenomic testing done in a number of different capacities. I've represented 3 here. One is in our pharmacogenomics clinic. One is in a population genetic screening, which is not just pharmacogenomic alone, but it's also looking at cardiovascular risk and cancer risk. And then the last one here is any clinician in our system can order pharmacogenomics for a patient. But really central to all of this is this middle point where all of these tests and test results are fed into our electronic medical record and then supported with CDS to help maximize the value and create a scalable solution across our landscape. And then we've created the safety net of our pharmacogenomics team to help fill in the gaps where the CDS doesn't do its job. So CDS is really key to what we're doing at North Shore. Let's go to the next slide, please. So what does CDS look like? Well, clinically, we're supporting more than 22,000 patients. We're supporting 20 genes linked to 70 different drugs. Our types of CDS include interruptive alerts, a passive portal. We use order sets, especially in treating depression as well as in treating pain. We have documentation tools where we can very quickly write a consult. And then we put interpretive reports for everybody who gets a pharmacogenomic result at North Shore. And just to give you a little bit of data, we really restrict down our interruptive alerts, so we're not triggering too many of those. It's really the most severe interactions are going to drive interruptive alerts. And last month at North Shore, we had 49 interruptive alerts. We had it overridden 62% of the time. I'll comment on that in a second. Note that that's an improvement from where we were last year, which was 80% of the time. But for anybody who's familiar with the interruptive alert literature, something over 30% action rate is actually pretty good. And this is one of the biggest problems about interruptive alerts is understanding what are all the features you need to evaluate to make it clinically relevant and the most clinically relevant thing for that patient at the time. And so it's an area that we need to improve and think about integrating genetic information with other clinical information so that you get one appropriate answer for a physician that's not overridden, well over 50% of the time. Okay, we'll go to the next slide, please. So in conclusion, CPIC is an accelerator to get people to implement pharmacogenomics. Those guidelines are crucial to what we do. I use them every day in my clinical practice. CDS is a requirement for a successful system-wide deployment of pharmacogenomics. I can use pharmacogenomic data on 22,000 patients on a daily basis because I have CDS in my system that works across the whole system. So no matter where you get testing done, you're getting the same level of clinical care. Discrete data is a must if you're going to do clinical decision support. There are many types of CDS. Pick the right one for your clinical situation. And again, interruptive alerts are not enough. Don't stop there. Keep going, drive, come up with the best solution for you, your patients, and all of the clinicians that you're serving. And I thank you for your time.
Thank you, Mark, for sharing this very exciting and important work. So next we'll move on to Dr. Stephanie Huang, who is a tenured associate professor at the Department of Experimental and Clinical Pharmacology at the University of Minnesota. She is also an associate director for the Institute of Personalized Medicine and Pharmacogenomics of University of Minnesota Alliance, and a member of the Masonic Center Cancer Center at the University of Minnesota. She is a chair of the International Pharmaceutical Federation Special Interest Group on Personalized and Precision Medicine, is co-organizing this webinar series. She serves on the editorial board of Annals of Translational Medicine and Frontiers in Pharmacology. To date, she has published over 90 peer-reviewed research papers, many of which are in high-caliber journals. For example, Nature Medicine, Blood. Dr. Huang is a board-certified clinical pharmacologist with extensive training in genetics, molecular and cell biology, clinical trials, and high-throughput data analysis. So now I'm going to hand over to Stephanie, who's going to be talking about precision medicine approaches in a key disease category, so in cancer. So I'm very excited.
Well, thank you, Donnacha, for that kind introduction. And good morning, good afternoon, good evening to all of you. And thank you very much for being here today. So as Donnacha mentioned, I am going to, in the next 15 minutes or so, give you an example of application for precision medicine in the field of oncology. Why oncology? Let's look at this figure showing here is an article looking at in the past 20 years, basically from the year 2000 to year 2020, all the FDA approved new drugs in the market, and how many of those has also biomarker. Many of them are pharmacogenomic markers in their drug labels. Right away, if I can direct your attention to this pie chart, this blue area is representing oncology drug. Almost 50% of the newly approved oncology drug in the past 20 years have had biomarker in their drug labels. And when you compare to the second largest pie chart, that's for the neurology area, that's only about 9% of the newly approved drug in the past 20 years. So this is really highlighting in the field of oncology, there's no way to get around about knowing biomarkers for the drugs if you're dealing with all the drugs coming into the market. The bar charts here, it's break it down into years. Again, the red areas representing those oncology drug versus the blue section are for non-oncology drug. Once again, you can see a large portion of reds are presented from the year 2000 all the way to 2020 with almost an increasing trend. More and more oncology drug has been approved with biomarker listed in their label. So this next slide, please. Seeing that there's clearly a lot of pharmacogenomic marker related to newly approved oncology drug. Why is that? These are some of the reasons that you can think of. First of all, cancer is a genetic disease. Because all cancer are derived from healthy normal cells with additional genetic changes, it's not surprising we're looking for differences between cancer and normal with the goal to find out compounds and drugs that could specifically targeting these differences. And to be honest, right now, looking at the US hematology oncology practice, genetic profiling has become a routine practice for majority of academic institutions, as mentioned by Mark, like community hospitals and other places are taking on these genetic profiling as well in their disease management. And if you are in the field of cancer treatment, or even not, like when we think about cancer therapy, we knew that many of the drugs that we use tend to be toxic. So there is an issue associated with narrow therapeutic index. So many of the drugs we use do not necessarily work in patients. So they have relatively low response rate compared to many other efficacious drugs in other therapeutic area. And equally important, we knew a lot of the anti-cancer agents we use are associated with high frequency of toxicity, and oftentimes those are very severe, could be life-threatening. So we really need to get it right, both to save the time for a fast-progressing disease to make sure you capture the best time to treat these patients, prevent the tumor from growing. At the same time, we also have to really spare patients from severe toxicity and protect them from over-treatment. So these are very important reasons for why we're interested in and needed to come up with things to select the patient and give them the right drug and right dose. A third reason I would say is because, again, it's somewhat of a luxury because in the field of oncology, we have many different drugs that has been proved in the recent years. So having that option, if you have certain genetic contradiction for example, if a patient come in with a genetic marker that was associated with severe toxicity, we can have the luxury to switch an agent, choose something else, versus if we only have one agent to choose from. So these are some of the reasons I believe that for the pharmacogenomic to be so prevalent in the field of oncology. Next slide, please. So one extra thing to keep in mind, if you're dealing with cancer and oncology, what Dr. Donnacha talked about early are mostly related to the germline genetic variation. So those are what I show on the left panel. So these are the things, simply think about it, is associated with host DNA, the normal cells DNA. So these are in general heritable, meaning it can be passed from parents to the offspring. And once it's passed, all cells in the person's body is affected. So every single cell will carry the same type of genetic variation. And these kind of genetic variation could impact both the pharmacokinetics as well as pharmacodynamic of drugs. However, in the field of oncology, there's another layer of genetic we have to consider, which is the somatic mutation. So these are additional genetic changes based off these germline genetic variation. So that happens only in the individual's body. And these are in general believed to be acquired and not heritable. And for the most part, this is unique to the individual, and it could affect both PK and PD. But when we think about therapeutic strategy, for the most part, we're associating these additional cancer genetic changes with pharmacodynamics. Next slide, please. So looking at how we have been applying pharmacogenomic in precision oncology, right away there are about these four bulleted area that I can think of. One, the top one is associated with disease prevention. So those, again, if we are considering two genomic layer, these are mostly associated with germline, the host DNA variation. If a person was born with certain higher variation for certain gene, in this case, one example I can give is the BRCA1 mutation. These, by carrying variation in this particular gene, an individual would have higher possibility of having breast cancer. So in this case, predetermining these genetic variation in their germline DNA could help people to make decision on how they want to go about prevent cancer from happening by lifestyle change and other things. The second, third, and fourth bullet points are more related to therapeutic strategy. So one is about drug metabolism. Again, this is more from the germline point of view. A genetic variation happen, for example, in drug metabolism gene could impact both the efficacy and toxicity of a drug. In the efficacy front, for example, if the drug is a anti-cancer agent is designed as a pro-drug, these metabolism enzyme could result in higher amount of active metabolites formation, therefore lead to higher efficacy. At the same time, if a germline genetic variation present in a key metabolism enzyme, which is breaking down the drug, and that could also lead to, so the variation, for example, it would lead to lower activity of that metabolism enzyme. Therefore, there's less breaking down of the drug. Therefore, there could be more like accumulation of active drug in the body, which could lead to higher toxicity. In the field of cancer, one area, which is the third bullet point, it's about the somatic genetic mutations in cancer. These has been heavily studied, and many of which has been associated with a new drug approval as a companion biomarkers. For example, this BRAF certain mutation in the BRAF gene, it has been indicated for response to BRAF inhibitors. So oftentimes in the recent years, when a new drug was approved in cancer, people are looking for specific genetic change in the cancer. And because the drug is designed towards that change, so only when the patients are determined to have that mutation are sort of would be indicated for that treatment. The last bullet point is about, when we think about cancer as a complex disease, even though many of the drugs we design are targeting one specific things, a very complex disease often are a result of multiple genes. So they're also approach focused on not so much on the DNA level, but rather on the RNA level, looking at tumor gene expression and collectively and using that as a way to determine the prognosis of disease as well as therapeutic strategy. So next slide, please. Because this is a technological stage, so I want to at least make sure that when you are thinking about these things, you are aware of, you don't have to memorize any of these things, but I think it's good to know these are the technologies that is currently available, has been used in the clinic in order to conduct pharmacogenomic testing in oncology. Oftentimes we refer things into a gene, but a gene could mean many different things. We could mean on a DNA level, a genetic change, and even on that term of genetic change, it could mean DNA mutation, copy number changes, structural arrangement, and all that thing. And a gene could also refer to RNA level, and it could also refer to protein level. So it is good to know if you order a test or having a test coming your way, whether they're measuring patient DNA level or RNA or protein. And also these different tests, the bold one listed on the slides are the ones that's already in the clinical practice. And the one that was not highlighted are also often used in the research realm and possibly coming into practice in the future. The other thing to keep in mind with technology is that we can really do things on different scale. So we tend to start with looking at one variant or one mutation. And nowadays, whole genome with the next generation sequencing technology become available, we can really do this very efficiently by looking at multiple things all at once. And so that would significantly shorten the time from sending the sample to getting a result, oftentimes measuring hundreds of different genes, mutations, or expression level. So this is just for your information, but I think it's important to know these, the presence of different level of measurement, and in order to better interpret a result. Next slide, please. So because of all these different use area, I want to just focus on maybe two of those. The first one is related to somatic mutation. Next slide. Usually the current, because we're talking about current practice in the clinic, this is sort of an example in the US. So usually as I mentioned earlier, many of the academic institutions now has been employ genetic profiling as their routine patient care for oncology patient. So the patient will come in, their tumor will be biopsied and sent for sequencing or genotyping, either through in-hospital laboratory, which usually through pathology, or outside through like Foundation One or other commercial source. And then the report will come back. And then the management team will come together to form a molecular tumor board, which consists of a multidisciplinary team, oftentimes hematology oncologists, pharmacists, pathologists, nurses, and geneticists, and others. So they come together looking at the report from a number of patients to look for basically association between reported genetic changes and available drugs to treat this patient. If So maybe we can go to next slide because there are many. So this is in this chart, I'm showing one of the website provided and updated by Memorial Sloan Kettering. So this is called oncokb.org. Basically, they're listing all the different level of evidence supporting the relationship between pharmacogenomic or genomic biomarkers to different drug in oncology field. Basically, the higher the level 1 and level 2A and 2Bs are things that could right away be used in the clinic. So level 1 is really straightforward. These drugs are FDA approved to treat the disease, and also the biomarker was approved by FDA as a companion biomarker towards that drug. So that's no-brainer. If a patient report come in with the mutation indicated and approved by FDA, and the drug is indicated to treat that disease, right away the patient will be put on that treatment. Level 2A is somewhat similar, with the exception that it's not the biomarker may not be approved by FDA, or maybe sometime the drug is also it's used. Let's say it's used to treat breast cancer, but in the case of patient, we are considering lung cancer. But the evidence has been very strong towards the relationship between biomarker and the drug. And level 3A is getting to the investigational implication. So there's compelling clinical evidence. However, they're neither approved or being clinically tested for. So this is almost like in the practice, the off-label use. There's strong evidence towards it, but there hasn't been vetted through a clinical trial. Level 4 is more of on the research level. There's hypothetical indication. So if you have a chance, you can log on to this website to see, but that really gives you a good evidence about what we know of in the field of oncology, how many genes and what drugs can be used for a certain therapeutic area. Next slide. So beyond the somatic gene changes and their quantification, another collection of things, as I mentioned, are gene expression signatures. Next slide, please. So basically, instead of measuring genetic mutation one gene at a time to determine what drug is associated with that gene, this is measuring a number of genes on the expression level, and then use that collection of signature to decide what is the probability of prognosis impact of those tumor. And then if it's for example, Oncotype DX is one of the example they use for breast cancer. They measure about 20 different gene expression in the tumor, and collectively with a mathematical equation, and the tumor will be put on a score. Basically, this is a disease recurrence risk score. So the higher the score, you're more likely to have 5 years disease recurrent. Therefore, you might need to be treated more aggressively because the nature of your tumor. So currently, there are many of these different commercial tests available in the field of breast, colon, lungs, prostate cancers, and also melanoma. So, and the list keep growing. Let's go into the next slide. So basically that brings me to, this is a really fast way to give a sort of capture of the current practice in the field of precision oncology. I am really excited for all this happening. Really, I think the incorporation of precision oncology has changed the practice of oncology really profoundly. And that, to see that to manage cancer patient nowadays, genetic tests become more and more of a routine, and we can make decisions based on rationalized science and all that. That's very, very important. However, that's not without any challenge. For example, number one, I would say patient expectation. So because of that proposed possibility, now we can tailor your tumor changes to a drug. Patients oftentimes are expected to, if I get my genetic tested, why can't you find that magic drug to treat my disease? If you had a chance to look at the website before, even though we have made significant progress on these, but as of now, there's only about a handful of genes that we have evidence that support their role. So think about human genome. We have about over 10,000 genes. Really, currently we have for about, like 20 or up to 50 different genes that we have drug targeted. So oftentimes the patient will send their tumor out for testing and the result come back. It's sort of, we do detect some changes. We don't necessarily know what that immediate matching therapeutic strategy is, which that leads to many different possibilities for clinical trials. So there's a lot of effort into quantify patient genetic profile and more efficiently assign them into clinical trial to test new drugs. And also from the flip side, not only this is patients' expectation, clinicians need to really keep their knowledge up to understand this ever-growing list of new drugs and new genes. This is particularly important for us as pharmacists. The list just keep on growing, and knowing the complex of genetic of human being and cancers, there are just a lot to keep up with and learn about. And also in order to really efficiently integrate that into the practice of helping patients. And cancer is not a uniform solid piece, and it's the cancer heterogeneity has been evidence in the recent years, and also there's drug stimulated evolution and other way of evolution. So the treatment strategy need to be changed. And nowadays with next generation sequencing testing, a large amount of genomic information become readily available. How can we keep that? How do we manage these data, put them in the safe place, and also efficiently use them, sort of related to what Mark just talked about. How do we, if we do whole genome sequencing in the future, how do we pick and choose information or make them useful in the clinical realm? And of course, there's a treatment and accessibility issue. I'm talking about the example of current practice in the U.S. World is not always the same, so we really have to adjust for local practice environment and decide on how we can best apply these knowledge towards everybody and make sure fair accessibility for everyone. Of course, there's payment reimbursement issues as well. And my last slide, next slide, please. Thinking about what's the future, maybe just a plug-in for the next session. One thing, I'm sure if you log in for our next session, you'll hear about there's new technology. What I presented today, getting biopsy from the actual tumor, doing the genetic profiling, but there are many new ways that has been launched and studied. For example, the circulating tumor DNA, circulating cells. These are making sampling of tumor or sample much easier than those more invasive strategy. And then the turnaround time for these genetic testing has been improved, and there's many new technology has come into play to make this really real-time, or at least near real-time detection of genetic changes in order to help making clinical decision. There's also additional model has been built, for example, getting cancer cells out of the patient and doing sort of on the Petri dish kind of culturing and exposed to many different drugs in order to sort of using patients' own material to determine how they would likely respond to various medication. And then gene therapy, immunotherapy, and all these things are coming towards precision oncology, which are all very exciting. I believe this is my last slide. Let's go to next. Well, thank you very much for listening.
Thank you very much, Stephanie, for the very insightful and comprehensive overview. So next, I'm going to introduce Einar Moltzen. So Einar is a medicinal chemist by training and has for almost 30 years been working with R&D at H. Lundbeck in different managerial as well as strategic positions. In 2016, he became independent and founded Somami Consulting, APS, which is a consulting company focusing on providing support within R&D projects, strategy and policy initiatives, collaborative contracts, and project evaluations. He is supporting public agencies as well as private enterprises nationally as well as internationally. He's affiliated with the Innovation Fund Denmark, where he is agency's lead representative within international activities relating to personalized medicine. He is currently elected as chair of the International Consortium for Personalized Medicine, so ICPERMED, which is an EC-funded European initiative to promote implementation of personalized medicine. And this is why he is here to talk today. So to give an overview of ICPERMED and also international coordination efforts in precision medicine, which are crucial to its success. And now I'm going to hand over to Einar. Looking forward to your talk.
Thanks a lot, Donnacha. And thanks a lot for giving me the opportunity to be here and present our organization. And what I will do is to talk a little bit about who we are, so you understand where I come from. And then I will provide a somewhat different perspective than the previous talks, because I would like to kind of give a more overall perspective on where we see what the status of personalized medicine or precision medicine is today. So if you go to the next slide. As the name says, we are an international consortium. Our main bulk of our members are members from the member states of the European Union, but we also have, as you see on the map, a number of international members. And what we try to do is to promote development and implementation of personalized medicine over the full value chain. And we are working not at a kind of an active research perspective or funding perspective, but we are kind of working from a more policy communication perspective. And what is interesting about our organization is that our members are mainly coming from national funding agencies, national ministries of health, and also non-for-profit health research funding agencies. So through our members, which is almost 50 now, we have a very direct access to decision makers and funding organizations, and there we can try to align what our members are doing in the different regions and the different countries. Our secretariat is funded by the European Commission, and we were started in 2016 and will continue until 2023. If you take the next slide. So besides our internal activities of networking, we also do another more outgoing activities. So we have every year we have either a conference or a workshop, and we will have the next workshop in June. We also have published a couple of key papers, which I can recommend you look into. The one is the action plan, which we developed back in 2017. And it's basically a roadmap on everything that needs to be addressed in order to develop and implement personalized medicine. And although it's about 5 years since it came out, it's actually very relevant still. And then a couple of years later, in 2019, we made kind of an update to see, what is our vision for personalized medicine in 2030. And then we have a number of other activities. I will not go into detail here, but I can refer to our homepage and try to look into the other activities we have. If you go to the next slide. So we are not alone. So we are kind of the umbrella organization, but there is a whole range of other organizations, the CSA's communication and support activities, which are all funded by the European Commission. Some of them are they're all trying to promote personalized medicine. We have a couple that is promoting into regions in Europe. We have a couple that is reaching out to China. We have one reaching out to Africa, one reaching out to Latin America and Caribbean. And then we also have some more kind of technical focused CSAs, the PERMIT, which is trying to develop new clinical strategies for development of personalized medicine, and HECO-PERMIT, who is looking into health economy perspectives. And then there's all the green ones, which are funding activities, and particularly ERA-PERMIT has been very successful over 5 years, funding a lot of good projects within personalized medicine. If you take the next step. Next slide, please. And I don't need to go into detail here, because why do we do all this work? But I guess we all know here that there's so many good arguments to develop personalized medicine, and by the end of the day, it will provide a much better treatment and much better health conditions for our patients and our citizens. So if you take the next slide, please. There has been a lot of interest in the area, and we were very lucky that very early on, and that's about more than 10 years ago, the European Commission kind of saw the light very early. So they saw the importance of this area and needed to develop it. So over these last 10 years, the European Commission alone has provided a budget of more than 2 billion euros in order to develop personalized medicine. And this is now also going into regional and national arenas. And we see now a lot of activities of national activities, regional activities, and also cross-national activities, which is also working on developing personalized medicine at all levels. Next slide, please. And it's kind of interesting to see this development from another angle, because when it started out, it was basically focusing on genomic research mainly, and to see whether these insights could provide better understanding of the disease pathologies. It then, as we also heard in this talk, has developed into how these findings can provide a basis for treatment recommendations. And what came later on, even though, and Mark alluded to that point also, is that it's not enough just to look at biomedical and genomic data. We also need to have the other healthcare data available. So kind of the doctor-patient interaction here is extremely important in order to really get the full picture of the individual patient. So this is not so advanced today, and but it's developing. And the next thing which is going on also now is to implement these PM-based methods into our health systems. And I'll come back to that a little later. And not too far ago, we started to discuss, and this discussion is increasing, about personalized prevention. And I will not touch on that, but it's a very interesting area, which will be very important going forward. Next slide, please. And this is a nice example from the France Genomic Medicine Initiative. And they are trying throughout France, through all the hospitals, to set out kind of a system where all cancer patients can go through a very structured process, starting from the first consultation with the doctor and taking all the necessary tests on the way and getting all experts around it together in order to come out with the best possible treatment conditions. So this is something that is well underway. So next slide, please. What we do in IC Permit is that we once in a while, we take a step back and then we see, where do we need to focus now? Where do we need to push the PM agenda in which areas? And the 5 focus areas we have selected for about a good year ago is on clinical studies in personalized medicine, PM in healthcare, patients empowerment, education and curricula in PM, and health economic value of PM. And in that regard, I can say that we have our next workshop in June, which will focus on ensuring value-based implementation of PM. And you might ask, why are we focusing on these areas? Because if you go further back, our main focus was basically on more preclinical basic research in PM. And if you go to the next slide, I would like to talk a little bit about where do we see where PM is today. And in a very simplified manner here, I mean, where are we in our implementation of our vision of personalized medicine? And I would like to address kind of from 2 points of view, from the research and innovation point of view, but also from what I under one hat is calling the policy point of view. And let me take it step by step here. So let's first take a look at research and innovation on the next slide. So if you look at where are we over the last 5 years within personalized medicine in Europe, there's a lot of support from the European Commission. As I mentioned already, there's a lot of national and regional PM programs already on the way. And we also see now that industry is also seeing the light here and realizing that this is the way to go. And what I did not say before is that maybe the most important development we have had in personalized medicine is the realization that personalized medicine has become a key component of, it has been realized as a key component of our future health systems. If you go back 10 years, it was kind of more a scientific oddity, you might mention, and at some point people said, well, this could be a kind of a boutique access where the most wealthy could get some good treatments. But now it's actually been realized as the most a very important component in developing our future health systems. There has been, as we already have heard now, a lot of new RNA developments. That's very good. However, the focus is still on cancer and also now on rare diseases. And that is good because there's a huge need for better cancer treatments. And there's also some good progress. But what I would like to mention here is that it's still only less than 10% of all cancer patients who are treated well with PM-based treatments. So there's a need for a lot more research. And you might ask, why is it not better? And I think also Stephanie alluded to that. And that is that, well, why can we not do it better within cancer? Well, one thing is that it's true that it's a genomic disease, but very often we don't have the full genomic picture. It's basically very rare that it's a monogenetic disease, so there are multigenetics in place. And the other thing is, of course, the lack of effective drugs. Because one thing is to identify the defective gene, but how it then affects the disease pathology and where you need to interact with this pathology, which target, is a huge task to identify and then to find the good drug. So this is also a major problem. But still, I mean, cancer is not the only problem for society. We have all the other big killers of diseases, and we need to go also the PM way in these areas. And there we are, unfortunately, not very far today. Next slide, please. Some words on the policy aspects. One thing that is important and also has been mentioned by Mark here is the availability of the necessary data. And here we both need biomedical, but also healthcare data. And they are needed both in order to do PM research, but also to develop the best possible treatment for the patients. And there's a lot of activity, local activities, and Mark was a very, very nice example on how to pull together data and how this can be used to the benefits of the patients. However, if you look at it from a broader perspective, it is still very problematic. And one of the major problems is that if you want to share data, I mean, we don't have simply the legislative frameworks necessary in order to ensure privacy and security of data access. And here we run into ethical, legal, and society also problems in a broader aspects. There are some activities going on in order to solve this. In Finland, they have come pretty far with where the government actually have put into place a legislative framework that can help this on the way. But this is only one country, and a lot of other countries are not really that far. We have seen, I showed you the French example, but also the NHS Genomic Medicine Services in UK are pretty far in trying to set up a system to collect the data. There's still problems in order to really get it out and using the data to the benefit of the patient. And Denmark is an example where the government has taken an overall strategy decision to implement personalized medicine, but is currently struggling with getting it into play and getting it to work, mainly because of these bottlenecks around the legislative frameworks. And then there are fortunately also some European initiatives, like the 1 Million Genomes Initiative and the Beyond 1 Million Genomes Initiative, as well as the upcoming European Health Database, which is a commission-driven activity. And they are also trying to support and to create the framework needed in order to share data, not only regionally and nationally, but also cross borders. And there we have some real problems. And one thing that is really also is a challenge in this point is the need for real world data. Going forward, we cannot only rely on data coming from clinical settings. We need to have the data, how it works out in real life. And in particular, if you go down to very small patient groups, there's a need to have you need to go out in the patients directly and get the data there. And one thing, if you also go outside the specialist areas, for example, in cardiovascular disease, metabolic diseases, and so on, a lot of the data is something that we should have directly from the patients through apps and other things. And these frameworks are definitely not in place yet in order to really exploit these data and use them. So there's a lot of work to do here, and this is on the policy level, and this is also what we try to do in IC Permit, is to push the politicians and the decision makers in order to see and realize these needs, so that the science and the research can have the framework that is necessary in order to really develop PM. Next slide, please. And then on my previous slide, I had two other things. One thing was around health economics. And this is really one of the big things right now. And I could talk a very long time about that. But I know that you have an upcoming webinar on the cost of PM. So it will be discussed there. So I will leave that here and just talk a little bit about implementation of PM in healthcare. Because this is another thing where also the decision makers maybe do not really realize how much is needed in order to transforming our healthcare systems into something that can adapt PM possibilities in the best possible way. And one example, it's maybe not so, besides the health economic problems, it's maybe not so difficult seen from a specialist area like cancer. But if you go into the more DP-driven indications, as I mentioned, metabolic diseases and so on, the main interaction between the patient and this health system is through the patient's GP. And to have really the DP realizing and learning about the possibilities and the tools he can get from personalized medicine is a major task. And we need to have a health system that can take care of this part as well. So we will have to look forward to some reforms in healthcare systems. Next slide, please. So I said that our organization will end, and also all the other CSAs will end in 23. So what then? And here I will just mention that we are now working hard on setting up the European Partnership for Personalized Medicine, which is a new partnership under the Horizon Europe umbrella. And it will be about a 7 to 10 year initiative. And there's a lot of interest for all the member states in Europe. And there's also a lot of international interest in this partnership. And this partnership will take care both of, it's indicated in the blue circle, so it both of funding activities, which both will develop new treatment possibilities for the patients, but would also include demonstration projects and pilots to show the regions and the countries how they can implement these things in their health systems. And besides that, it will also include all the policy activities we are doing right now in IC permit in order to push the political agenda in order to promote personalized medicine. And of course, we will work closely together with the other 8 partnerships that are on the way. And I think I'm coming to my last slide. Yeah, if you want to learn more about it, I can refer to our homepage. We also have a stakeholder forum. Everybody can become a member, and there we'll reach some more information. And we also have, as I said, further information on our homepage. And next slide. Yes, so thanks a lot for your attention. And yeah.
Thanks, Einar, for this very important and insightful overview of the policy side of implementing personalized medicine. So if I could, we're moving on to the Q&A section and panel discussion. So if I could ask all the speakers to turn on their video. And also for attendees, if you have any questions, please feel free to write them in the Q&A chat box or Q&A box. Okay, so now we can see everyone perfectly. Perfect. Okay, so maybe I can start first with a question. So many different barriers so even though there's been many advances on the scientific side for precision medicine in recent years, and you all presented some nice examples of its implementation, one could argue that it has not been so successfully and deployed widespread geographically and across disease areas, as Einar had mentioned. And some of the bottlenecks were discussed for this. But if each of you could pick one bottleneck that you think is the major bottleneck that the pharmaceutical community should direct its attention primarily towards. Maybe I can start with you, Einar, as you just discuss this.
I'm just thinking because just mentioning one is, well, I would still say kind of the data issue is a problem. I mean, it's not really developing, and it's as I said, it's not only a matter of sharing biomedical and genetic data, because from a scientific and technical point of view, that is not easy, but it's doable, because the format of such data are in a sense in a way that you can easily align them, for example, across borders. But still we have kind of the legal issues around sharing these data and patient privacy. But the other thing is on the healthcare data, because if you want to have the full picture around the patient, you need also to have the healthcare data available, and they also need to be there for research purposes. And they are very localized, because here we are maybe talking about going into and reading patient journals and extracting data of that sort, and to have these kind of data on a more global scale is a major challenge. So there's a lot of need for these things. So I will stick to that one for your question.
Yeah, I can definitely testify that this is even a huge challenge in the research context as well. So Stephanie, maybe you can also address this question.
Yeah, this is an easy and hard question. Like Einar, my mind is racing. I'm coming up with all sorts of answers. But I think one of the question in the question box, Miki mentioned about the standardized language or system. I think that's one of the barrier. Honestly, Mark sort of showed that, PDF is not PDF, it is not PDF. So some kind of standardization across the system, like the world in this case, would really help. I mean, even, you think CPIC or Dutch guidelines are one of the two go-to thing when we think about that in the pharmacogenomic world. Even those two top-notch figures had, in practice, we often had the debate about there's discrepancy between the two guidelines, which one do we go to, how do we, that kind of thing. I think it's a hard thing to do because there's we're trying to strike a balance between a complex system to a uniform, sort of generally uniform answer. So I think as a start, if we could start gathering information in a somewhat uniform terms or knowledge. I know in the medical field, pathology has started leading the effort to come up with medical new term dictionary. So use the same term to describe the same problem. That could be a way to start, at least to look at the problem in a standardized way.
Yeah, for sure. And I also think that with this OncoKB approach, it would be very nice if this was maybe more widespread deployed across different disease categories. For better evidence-based decision making. And now, Mark, if you could also answer this question, that would be great.
Yeah, so I'll definitely acknowledge the other answers. They're huge problems, but I'll put a little different spin on it, and that is there's not a defined value of personalized medicine. And without having a defined value of personalized medicine, like, why are we really doing this? Like, what's the good of it? Everything else after that, like data and how you share the data, well, it's probably determined on what's the defined value of what you're trying to do. What data do you need to have? What's the nomenclature you're going to use? Is all central to this idea of what's the defined value? What's your defined use case? Why are we doing this? We've been having discussions about the value of personalized medicine since the 1950s, when the term pharmacogenomics was first coined. And the discussion has always been very similar and hasn't changed a lot. And I would argue that part of why that's the case is not enough people are implementing to actually figure out what is the value of doing this stuff. And so I'll put this out there to the group. Don't let perfection be in the way of progress. Try to start implementing at your institution at whatever it looks like today, what you can do, what's scalable based off your resources. And from there, do it in a way in which you can track it and begin to define the value for your institution. And then let's share that value and all come to a common understanding of why this is good and how this is going to impact and improve humanity going forward in medication therapy.
Okay, thanks very much. I see Einar raised his hand. Do you want to add
I
to this?
simply can't help myself because Mark is touching on my pet subject, and that is exactly the value of personalized medicine. And there has been, as I mentioned, we have had a CSA, a kind of a project that has been dealing with where we have had health economics expert trying to look into models for, is it worthwhile to do personalized medicine? And they made some very nice scientific models, but the problem is that the data they need in order to make it valid for the real world, so to speak, is simply not there. So they have to restrict their models to settings where they come to a conclusion where PM is not really worthwhile. But this is, in my opinion, simply because they need to take the full picture into play. And we hear a lot about these things. Oh, this cancer treatment is costing $2 million for one patient. We cannot afford that. But if you can turn that patient into a healthy and productive citizen who is productive for the next 30 years, I mean, this is a very small investment in order to do this. So we're missing some of these, there are other examples also, but some of these more broader perspectives in order to show the world and the politician and decision making that this is worthwhile. And the only way we can do it, and you mentioned that, Mark, is, and what we also try to do in IC Permit is to show the good examples, to show that it works. And the only way is go out and do it and let the research and the science do the good job in order to show that it works. And then eventually, I think people will. But there's a whole other aspect also, which I think is very important. That is, when people are talking about value of PM, they're talking about cost effectiveness. And this is, in my opinion, a very wrong attitude or wrong thing to look at. Because maybe PM is more expensive than the traditional types of treatments. But by the end of the day, if you take the full value in, as I said before, it's definitely worthwhile. So maybe it's worthwhile to put more money into PM in healthcare And then the end of it, in a total for society, will be a major gain.
Yeah, perhaps precision medicine is actually still going through a little bit of a teething phase where it will get more and more ineffective as time goes on, and that will make the investment more worthwhile over time. Especially since you can basically subcategorize patients or stratify patients at any level, and there's so many technologies out there to do this. That, yeah, basically once we define how to do this optimally, it will be even more effective. Okay, so maybe just move on to some questions from the attendees. So one question, and this kind of relates, I think, also to the kind of importance of implementing it in education for clinicians. So should pharmacists learn more about data analytics to help develop personalized medicine or precision medicine? And I guess this is like a very simple question, but like, what would you think is the first maybe priority steps to do this? And do you think the infrastructure is already in place for this? So maybe this is more relevant to you, Stephanie and Mark. Do either of you want to start?
Yeah, I'll jump in real quick here. So the answer is it depends. So if you have access to resources that are experts in data analytics, don't waste your time becoming an expert in data analytics if you're a practicing pharmacist, because you won't have the time to execute the skills that you develop. However, if you don't have that resource, then knowing some of this information can be really helpful. But as a practicing pharmacist, the biggest key that I think here that you can do is figure out how to structure your job and your daily activities to generate high value data that people can look at and make big conclusions off of. You should be, everything that you do should be generating data that can improve your job in the future. And if you're not doing it that way, you should really go back and look at it and figure out how you can be generating data to improve the future care of your patients through your day-to-day activities.
Yeah, absolutely. I agree with Mark 100%. And also, I think that's somewhat related to, I see quite a few questions. People are wondering, as a pharmacist, what our role in this whole precision medicine era. I truly believe, precision medicine has become a hot term recently. We have been, as pharmacists, we have been tailoring pharmacotherapy and all that stuff. It just adding additional layers. So the world is moving, the therapeutic is moving. So we have to keep up with the knowledge. If that's related to data analysis, might be it. One thing I want to mention is a lot of information become digitalized, and there's so much out there. So if you're referring to gaining data analytical ability, mainly just to keep up with the world, keep up with the progression of available knowledge, I think that's a must. I mean, in my own practice, in my own career, I feel like if I don't keep up, I got left behind. And so that was sort of a survival thing, not necessary for making, and that also ensure that I can talk to people in the right terms, right information. So I think to keep up, it's important because there's just so much data out there and so much, learning ability. There's so many tutorial classes. If you are interested in this, feel free to learn those. I agree with Mark. Every job type, you have your specialty. So don't expect yourself to be the health informatic person or the person hardcore into doing the constructing the network and structure, but understand what they need. And I also really very much agree with Mark and Einar. For pharmacists, anything you do related to pharmacogenomic precision medicine, collect data, not just what you did, but what you did, how does that impact the patient? How does that impact the physician? How does that impact your own work? Keep track of that because you're doing your work anyway, but keep that information. Those are going to become valuable to you down the road. That's my 2 cents.
Thanks, both of you. I see Einar also raised his hand for this.
Yeah, I mean, with just a comment to this, because I also think that this thing about learning or teaching health professionals about these things is so important. And if you can take it from different perspectives, I mean, I mentioned the GP. I mean, if you ask a GP, most of them, they will say, well, I have been doing personalized medicine all my life. And of course, when he's sitting with an individual patient, he's treating the patient as an individual. So this is kind of the wrong question to ask you. And the thing is to say the GP should learn about the available tools that he can help his patients with. And this is one thing, and I guess the pharmacist is in a bit in the same situation. And here we are more dealing with not to run into a data overload, because I have seen a lot of projects now. They want to develop a website or an app or something which a GP can use. So they have an app for 5 different CV conditions, an app for 2 different metabolic conditions, so on and so on. So by the end of the day, we offer the GP zillions of apps, and he's busy with the patients. He doesn't have time for that. So we need to manage the data flow, and we need to put the data in the right format so it becomes a value for the GP and the pharmacist, and also for the patient, I could add.
Thanks very much. I do think, yeah, maybe it's an overkill for pharmacists to become professional programmers or professional data scientists. Yeah, but I think it might be useful. I mean, this is just from a pharmacist who transitioned to computational biology a few years ago. It is definitely an uphill battle to understand these statistical models. Maybe more coverage of these in future pharmacy education could be useful, although not necessarily becoming professional data scientists, of course. Okay, I think that actually brings us, we're already at time. So I just have a few more slides to wrap up. Thank you all for this panel discussion. Let me just return to sharing my screen. Thank you all for the questions as well, of course, very nice. Okay, so just so for some upcoming events. So this year, the FIP Pharmacy Practice Research Special Interest Group Summer Meeting will be held in person on 4th and 5th of July, 2022 in Utrecht University Applied Sciences, Utrecht, Netherlands. All PhD students and their supervisors, researchers, academics, professional organizations, and practitioners involved in research are welcome to join this meeting to build a global pharmacy network, present their research by submitting an abstract, and increase pharmacy practice contributions to global health. The program will offer interactive workshops and keynote sessions, networking events, and interactive closing panel discussion. So book your place now and register before 30th of June. And also we're excited to invite you to the 80th FIP World Congress of Pharmacy and Pharmaceutical Sciences in Seville, Spain. This Congress will take place from the 18th to 22nd September 2022 under the theme Pharmacy United in the Recovering of Healthcare. Registration and abstract submission are already open. For more information, check out the website seville2022.fip.org. So the important dates are for abstract submission deadline 5th of June, and the results will not be announced on the 1st of July, and there's an early bird registration deadline on the 15th of July 2022. Okay, and you can check all future FIP digital events at events.fip.org. And last but not least, thank you all for attending. I would like to especially thank the panelists for your very important insights. So much expertise in one event, which is very nice. I certainly learned a lot. So the recording of this episode will be made available on www.fip.org. You're welcome to give feedback to webinars@fip.org so we may continue to improve our digital event offerings. And thank you very much, and good day to you all.